There we go. All right. So good afternoon, everybody, and welcome to the Department of Medicine's Grand Rounds. My name is Paul Hahn. I want to thank all of you for joining us today. Next week will be our Kochman Lecture at Medicine Grand Rounds titled Sjogren Syndrome. This conference will be presented by Dr. Alan Baer, Professor of Medicine and Director of the Jerome Green Sjogren Syndrome Center at the Johns Hopkins University School of Medicine. We hope you will all join us for that presentation and discussion next week. Moving on to today's Grand Rounds. Today is our Institute for Clinical and Translational Research talk at Medical Grand Rounds. And today's talk is titled The Use of Visual Deprivation to Promote the Recovery from Severe Amblyopia, Translation from Animal Models to Humans. Our speaker today, who has graciously joined us from the University of Maryland College Park, is Dr. Elizabeth Quinlan, professor in the Department of Biology there at the University of Maryland College Park. Dr. Quinlan received her PhD in physiology at the University of Illinois, Chicago. She then went on to complete her postdoctoral training at the University of Virginia and at Brown University. Since then, Dr. Quinlan has gone on to conduct groundbreaking research on visual cortexes to investigate synaptic plasticity in the brain, with her projects at that time focusing on how experiences affect the juvenile and adult mammalian brains through visual system plasticity. Her current focus is on binocular visual deprivation through dark exposure and how that can potentially restore visual cortex plasticity and promote recovery from amblyopia. Her work has been recognized at a national level, and she's received numerous awards from organizations like the Neuro-Optometric Rehabilitation Association, and she currently serves as the Clark Leadership Chair in Neuroscience and the Director of the Brain Behavior Institute at the University of Maryland College Park. Please join me in welcoming Dr. Quinlan, and please feel free to type your questions in the Q&A box, and we'll make sure that Dr. Quinlan can see them at the end of her talk. So Dr. Quinlan, I will be stopping my share, so please feel free to share from your end, and then you can take it away whenever you're ready.
Great, Paul. Thank you so much. So I want to tell you about another role that I'm playing in College Park. As you may know, the Empower mechanism, Empowering the State, just recently awarded funding for College Park investigators to compete for some of the ICTR resources, ATIPs and KL2s and TL1s. And one of my roles here in College Park is to encourage and recruit investigators to compete for those awards in an effort to strengthen bi-campus collaborations and also to strengthen our effort in the translation of basic sciences to a human and clinical population. So today I'm going to tell you about a story, work in my lab, where we did just that, translated foundational work in animal models to a human population. And the title is The Use of Visual Deprivation to Promote the Recovery from Severe Amblyopia. So any of you that have struggled to learn a second language as an adult and seen the ease with which a child accomplishes the same task knows intuitively that there's something very different about the way the juvenile versus the adult brain acquires and stores and recalls information. And although there is presumed to be evolutionary advantage, a fitness advantage to stability over plasticity in adult circuits, there are also consequences. There are also negative consequences. And one is that the adult cortex loses the ability to recover from damage. One of the best studied models of plasticity that's confined to a postnatal critical period is the refractoriness to the recovery of amblyopia in human patients. So amblyopia, which is Greek for dim vision, is caused by an asymmetry in the quality of visual inputs across the two eyes. And I'm showing you here a child with a unilateral congenital cataract. The cataract is blurring the input into one eye, specifically at high spatial frequencies. And if left unrepaired, the vision in that amblyopic eye will be very compromised. this is in contrast to the vision through the normal eye, which will be normal. So there's an asymmetry in the quality of visual experience across the two eyes, and also a gross asymmetry in the spatial acuity across the two eyes. There are many ways to induce amblyopia. Misalignment of the eyes due to lack of coordination in the oculomotor muscles that control the eyes causes strabismic amblyopia, as is seen here on the top right. You can also induce amblyopia by having a large difference in the refractive index across the two eyes. So a strong eye and a weak eye will induce anisometropic amblyopia. And then the most severe form of amblyopia, which I've already introduced to you, which is induced by the presence of a unilateral congenital cataract, causes deprivation amblyopia. So in all cases, there's a difference in the quality of the input across the two eyes. The incidence of this type of asymmetry is very high. Some estimate as high as 6% worldwide. And all types of monocular amblyopia reduce acuity in the affected eye. They also share another characteristic, which is that they all are increasingly difficult to reverse with age. So in this case, I'm showing you data from an esotropic, a child with esotropic strabismus. Even after the strabismus is surgically corrected, the acuity in the amblyopic eye, shown in blue, is lower than the acuity in the fellow eye. And this is months after the after surgery and months after therapy in the weak eye. So it's very difficult to recover normal vision in the amblyopic eye. And here's a more dramatic example. This is individuals with deprivation amblyopia. These are child born with a unilateral congenital cataract. And on the x-axis is the age at surgery. The surgery is diagnosis and removal of a unilateral congenital cataract. And on the y-axis is the acuity, the spatial acuity that is eventually achieved in the previously deprived eye. So this is Snellen acuity. The authors of this meta-analysis, Mitchell and McKinnon, are Canadian. And so they're using a metric scale, but 6.6 here at the top is roughly equivalent to our 2020. You can see that there's an inverse correlation between the age at surgery and the spatial acuity achieved. And the horizontal line that I've drawn onto this graph is the legal designation of blindness. And so the cataract has to be diagnosed and removed very early in postnatal life in order to recover or obtain appreciable vision. So this is an example, of course, of a critical period. There are critical periods for the induction of amblyopia and critical periods for the reversal of amblyopia, much like there are critical periods of enhanced sensitivity to experience across all senses and across all cortical functions, sensory systems, language acquisition, and higher cognition. And despite the fact that the timing of these critical periods may vary based on domain, what they all have in common is that plasticity, or the ability to regulate function with experience, goes down with age. And this is due to the fact that synaptic plasticity, the ability to change synaptic function based on patterns of activity, declines with age. So we can model this in animal models. We use both rats and mice. We can show that plasticity in the visual cortex is lost with age by doing a very simple experiment. So we're going to use physiological approaches. We're going to surgically implant an electrode array in this red crescent here. This is the portion of the rodent primary visual cortex that receives input from both eyes. So this is the binocular primary visual cortex. And if we record from a normal binocular animal, and in this case, we're going to do a low pass filter, and we're going to acquire the LFP, very similar to an EEG recording. This has better source localization than EEG because we're recording at the source. And we're going to give a simple visual stimulus, and we're going to evoke a change in the LFP. So this is a visually evoked potential. In binocular subjects, same recording site, but stimulation given only to the contralateral eye, we get this large amplitude, beautiful waveform, this large amplitude visually evoked potential, same recording site, same visual stimulus, but given to the other eye, the eye that is ipsilateral to the recording site. We also induce a visually evoked potential, but much smaller amplitude. So this is part of the, this is one of the characteristics of the rodent visual system, is that binocular neurons, despite the fact that they respond to stimulation of either eye, still have a strong preference for stimulation of the contralateral eye. And this preference is shown here by the twofold difference in the amplitude of the response to contralateral eye stimulation to ipsilateral eye stimulation. So if we close one eye for 3 days, these experiments are done in young animals that are expressing robust plasticity. They're in the critical period, in this case, a 28-day-old rat. If we close one eye, if we close the eye that is delivering the stronger input, we get a decrease in the response to the closed eye. We get an increase in response to the open eye. So a couple more details. Our arrays span the full vertical depth of the binocular region of the primary visual cortex, and closing one eye in a young plastic rat for 3 days causes a decrease in the amplitude through the whole vertical depth of the cortex. So all layers of the cortex express this type of plasticity. We can do this simple ratio. We're just going to look at the amplitude of the VEP in response to the preferred eye, contra, over the non-preferred eye, ipsi. This is usually 2, which is shown in the gray bar. We see that in binocular animals. We close one eye for 3 days. We get a significant change in that ratio, significant decrease in the contra eye response, and increase in the ipsi eye response. But if we do the same experiment in animals that are just slightly younger, in this case, 70 days old, we see no change, right? So there's a critical period to recruit this plasticity. We can use a different physiological method to show the same thing. In this case, we're going to take our signal from our chronically implanted electrodes into a high-pass filter so that we can isolate action potential generations from single neurons. We can see at the single neuron level that these neurons also have a preference for contralateral eye stimulation. So if we look at the right, these raster plots, time is on the x-axis, and we're going to give the simple visual stimulation 100 times, 100 repetitions. So on the y is trial number. You can see that presentation of the simple visual stimulus evokes a robust response at about 150 milliseconds after stimulus onset, which is stronger in the contra eye than in the ipsi eye. This is in a normal binocular adult. And we can bin ocular preference based on strongly preferring contralateral eye stimulation, that's a class 1. Weakly preferring ipsilateral eye stimulation, that's a class 5. No preference is a class 3. You can see in binocular controls, this distribution is skewed to the left. Again, this is the expected contralateral preference, contralateral bias of the rodent visual system. We close one eye for 3 days. We change that bias. We shift preferences away from the closed eye, but only if we close one eye in juveniles, not if we close them in adults. So critical period, we would say, for the induction of this amblyopia-like plasticity. So the textbooks say that synaptic plasticity is lost with age, but there's always been a couple really interesting exceptions to that rule. One is that we know from clinical reports that individuals that have a mild to moderate stroke often spontaneously or with therapy recover functions that were lost immediately after stroke, aphasia being one of the most common. The neurons that are damaged or die during the stroke do not recover. The aspects of functions are remapped in the spared areas of cortex. That's the interpretation, of course. Here's some experimental data demonstrating a similar phenomenon. So the somatosensory cortex, neurons in somatosensory cortex have very, very small receptive fields, which means that if you're recording from a single neuron, in macaque, as the case is here in Tandon et al., if you're recording from a single neuron, you may only evoke enhanced activity by putting pressure or tapping on a very small area of the body surface. In the case of the green neuron, this little part of the palm, there are neurons that are coded here, red and blue, that respond to a very small area of a single digit. So the hallmark of mature sensory systems is very refined receptive fields that respond to very specific stimuli. But after deafferentation, okay, in this case, so after deafferentation, you get massive enlargement of receptive fields, followed by reassignment of receptive fields. Neurons acquire new receptive fields after deafferentation. So my interpretation of this work was that ongoing activity was limiting plasticity in the adult cortex. And deafferentation, if you remove the primary input to a region of cortex, you could unmask plasticity in adult cortical circuits. And so to test that hypothesis, we did a simple experiment. We deafferented the rodent visual system by putting adult post-critical period animals in the dark. So we're removing the primary input to the visual system. This was done quite a while ago. This is our initial publication introducing this phenomenon. All animals in this cohort received 3 days of monocular deprivation. They're post-critical period. So in control adult, the first data point on the left, monocular deprivation does not change the inherent contralateral bias of 2 of the FEP amplitudes. There's no plasticity, confirming these are post-critical period animals. But if we put the animals in the dark for just 3 days and then close one eye, we begin to see some changes in ocular preference. And if we put them in the dark for 10 days and then close one eye, we see a significant shift in ocular dominance that is indistinguishable from what we see in critical period juveniles in every parameter that we've examined to date. So this suggests that deafferentation of the visual system unmasks plasticity. So we want to know if our ability to unmask plasticity with deafferentation can be used to recover function in severe amblyopes. This is the Mitchell and McKinnon meta-analysis that I showed you before that shows that there's a clear critical period for the recovery from amblyopia. And we have studied this on many levels, but I'm going to show you the most important data set first, and that is spatial acuity. So can we use dark exposure in amblyopic rodents to recover their spatial acuity in their blind amblyopic eye? So to study acuity in rodents, we use a 2 alternative forced choice spatial frequency detection task. This is a water-based task, so a trapezoid filled with water, has 2 LED monitors mounted on the back end. One will show the positive stimulus. The positive stimulus is a high contrast sinusoidal grating. The other lane will show the negative stimulus, which is an equal luminant gray screen. Well, first, we train the subjects to associate the location of a hidden platform. So the water in this task gives some mild negative reinforcement to the subjects. And so they're motivated to seek respite from the water, and they learn to associate a hidden platform, which is not visible above the surface of the water, with the positive stimulus. So we switch this from the left to the right in a pseudo-random order. And once they learn this association, we put a divider in the middle of the tank to force a choice point so that we can calculate spatial acuity based on the animal's behavior at a known distance and the known spatial frequency of the visual stimulus. Once they demonstrate that they've learned the association, we begin to slowly increase the spatial frequency of the positive stimulus until performance falls off to chance, and we use that as an assessment of the subject's spatial acuity. So on the right is a frequency of seeing curve. Spatial frequency of the positive stimulus on the X, percent correct is on the Y. In this cohort at low spatial frequencies, wide bars, 100% of our subjects perform 100% correct choices at low spatial frequencies. This is extraordinary for animal behavior, and it shows that the trained rodents can perform this task very well. They understand what we're asking of them and they can perform it very well. Then as the task gets more difficult, spatial frequency increases, performance falls off. And the highest spatial frequency at which a single subject performs 60% or greater correct choices 2 days in a row, we use as our estimate of spatial acuity. All right, so that is the task. So now let's see what dark exposure does to acuity. So in these experiments, we're going to induce very severe amblyopia, the type of amblyopia that is induced by a unilateral congenital cataract. So this is an occlusion that's present before the optics clear and persists for very long duration. So in our rodents, we're going to do a chronic monocular deprivation with surgical lid suture at 14 days of age. This is again, eye-opening, but before the optics clear. This means that the subjects have no pattern vision in this eye. Then we're going to use the water maze to test their acuity, in their non-deprived eye. We get a measurement of about 1 cycle per degree, which agrees with many other labs. Then we're going to close the good eye and open the bad eye and test their acuity with their amblyopic eye. And you see that we have no evidence that the subjects can perform the task, even at the lowest spatial acuity with their bad eye. So they are functionally blind. But if we put the subjects in the dark and then bring them out into the light and then open up their bad eye and ask them to perform the task with their previously amblyopic eye, most subjects can perform the task at low spatial frequency within days of coming out of the dark. And we get a slow but complete recovery of spatial acuity following dark exposure. Now, if we introduce a delay between coming out of the dark and performing the task, we do not get recovery of vision. So the red data points show animals, again, chronic monocular deprivation, dark exposure, but then they were just put back in the animal colony for 5 weeks before we asked them to perform the discrimination task with their weak eye, and they could not. So this Eaton et al. Told us 2 really important things. One is that the reactivation of plasticity in the adult cortex by dark exposure is transient. And there's a window of opportunity when the subjects come out of the dark where instructive visual experience is able to capitalize on that plasticity and drive the recovery of acuity. We have documented many other deficits at the anatomical level, at the circuit level, cellular level, physiological level that are induced by chronic monocular deprivation. Not surprisingly, chronic monocular deprivation induces significant deficits in anatomical and circuit structure and function. Dark exposure promotes the recovery of every parameter we've examined to date. And here's just a couple examples of that. So again, VEP amplitude is normally 2. If we look at the ratio of the contra eye response to the IPSI eye response, that's significantly decreased by chronic monocular deprivation. It recovers to normal values, as is shown in the red following dark exposure. Neurons in primary visual cortex are exquisitely tuned to aspects of visual stimuli. This includes orientation tuning, which means individual neurons are only going to show an increase in their spike rate for visual stimuli that are at the orientation that they prefer. We can see that in controls. This is a post-stimulus time histogram. Time is on the X, repetitions is on the right, and you can see a very robust response in spiking input, increase in spiking output. when these neurons are given their preferred orientation, but not the orthogonal orientation. So this orientation selectivity is a hallmark of mature visual systems. We lose it with chronic monocular deprivation. We recover it when chronic monocular deprivation is followed by dark exposure and visual stimulation. And then I'll skip down to one anatomical example. We use dialytics to drive a green fluorescent protein into a subset of pyramidal neurons in primary visual cortex in order to look at dendritic anatomy as well as the morphology and density of dendritic spines that decorate, in this case, a basolateral dendrite of a layer 3 pyramidal neuron. You can see that they're spaced about a micron apart, and they have a lollipop-like shape with a large head relative to stem. Following chronic monocular deprivation, it looks like deforestation. The spines collapse, with the few spines that persist are thin and very immature. But following dark exposure and visual stimulation, we get a recovery of normal dendritic spine density as well as normal dendritic spine morphology. And dendritic spines, of course, are the site of excitatory synapses. So we're interested in two things. We're interested in understanding the mechanism by which dark exposure enhances plasticity, and we're interested in understanding if this is a general phenomenon. Can it be translated to other sensory systems, and can it be translated to other animals, including humans? And a lot of our mechanistic work has focused on a specific population of interneurons, the parvalbumin expressing basket cells. Parvalbumin is a calcium binding protein, which we can use to identify these specific neurons. And I'm not going to really talk about some of this mechanistic work other than to make one point, and that is we've identified two signaling pathways that seem to be unique to excitatory synapses onto this class of inhibitory interneurons, these parvalbumin inhibitory neurons. And we can manipulate these, anchor AMPA receptors to synapses. And so they play a very important role in stabilizing excitatory synapses and therefore maintaining excitatory synaptic strength. And we can manipulate either of these pathways, and we can increase or decrease plasticity. We can make adult visual cortex plastic. We can make juvenile visual cortex aplastic. So we just know how to manipulate plasticity in the circuit. But what's interesting, another interesting thing about this pathway is that these that have binding partners outside of the neuron in the extracellular matrix. As you know, neurons are embedded in a dense extracellular matrix, the composition of which is roughly equivalent to cartilage. And these parvalbumin positive interneurons have another signature in that the extracellular matrix around the neurons is especially dense. And I'm showing you here a plant lectin, WFA, that's complex to a green fluorophore, as well as an antibody against parvalbumin to show where these parvalbumin expressing interneurons are. And you can see that neurons expressing parvalbumin have a very strong condensation of WFA, a very thick and condensed, it's called perineuronal net around their cell bodies. So because we can control synaptic plasticity with these two signaling complexes, and because these two signaling complexes bind to entities in the perineuronal net of the extracellular matrix, we had a simple hypothesis. We hypothesized that when we put adult post-critical period animals into the dark, we degraded the perineuronal net, thereby reducing binding sites for these signaling pathways, reducing excitatory drive, and reactivating the cortex through disinhibition. And so now we're going to ask this question in adult mice. So I'm showing you on the left is a slice of mouse visual cortex going from the top through all layers down to the hippocampus. The green signal is this plant lectin that's carrying the green fluorophore, the WFA, and the red is the parvalbumin cell bodies. And this is a maximal intensity projection of several Z-planes. But you can see that there's diffuse staining, diffuse green throughout the vertical depth, as well as condensation around a subset of these parvalbumin positive interneurons. And if you look at the high mag, you can clearly see a perineuronal net around one of these parvalbumin positive interneurons. So then we put the animals in the dark and we did the same analysis and nothing changed. Okay? The results did not support our hypothesis. Dark exposure did not degrade the perineuronal nets, but interestingly, if we let the animals have just a couple hours of reintroduction to light, we drove down the extracellular matrix. We drove down the WFA signal. Surely this is clear to see qualitatively. The quantification is on the right. If we look at the intensity of WFA, which is reporting the extracellular matrix, we see in blue control the distribution of WFA signal along the vertical dimension. Dark exposure does nothing to change the WFA intensity, but light reintroduction significantly reduces it throughout the cortical depth, quantification of which is on the bottom. Importantly, we see a significant decrease in extracellular matrix density at both PV positive and PV negative pixels. So dark exposure followed by light reintroduction is degrading extracellular matrix everywhere, not just around the PV positive interneuron cell bodies. We've identified the enzyme that is responsible for the degradation of the matrix, and that's matrix metalloprotease MMP9. If we repeat the experiment in an MMP9 knockout mice, we see no loss of the extracellular matrix of the WFA signal when we bring a dark exposed animal out into the light. We can also acutely inhibit MMP9 activity with a relatively good inhibitor of MMP9. We're going to deliver that via mini pump throughout the 10 days of dark exposure. And if we do that, we block the degradation. On the right here, we block the loss of the WFA when we bring the animals out into the light. We can also repeat the experiment with antibodies against specific components of the extracellular matrix. These are lost when we bring a dark exposed animal onto the light, but not if we do it in the presence of acute pharmacological inhibition. So to ask where this is happening, we used a FRET-based biomarker. This FRET-based biomarker is a small peptide, which is a synthetic substrate for MMP9. On one end of the molecule is a fluorescent donor. On the other end of the molecule is a fluorescent acceptor. So fluorescence transfer causes fluorescence to be quenched. So when there's no enzymatic activity, cleaves this peptide and it interrupts the FRET. So if there's no MMP activity, there's no fluorescence because fluorescence transfer quenches the fluorescence on this peptide. And if there is proteolytic activity, the FRET is interrupted and we will see fluorescence. When we do this, we see an increase in, we see that MMP9 activity is punctate. It's a nice image on the bottom here. We get about a 1 micron puncta. The increase in area, the increase in the number, and there's an increase in fluorescence intensity. All of these parameters increase when we bring dark exposed animals out into the light. But then we can fix the tissue and lay on synaptic markers and identify, lay on neuronal and synaptic markers and identify the specific location for this increase in activity. And we see that the increase in MMP9 activity that is induced by light reintroduction after dark exposure localizes specifically with a marker for thalamic synapses onto cortical neurons. So dark exposure followed by light reintroduction increases MMP9 activity that is perisynaptic and is highly localized to thalamocortical synapses. So we're in the process of identifying the targets of this enzyme. As you know, here's an electron micrograph of excitatory synapse. Transsynaptic protein bridges hold the pre and postsynaptic contribution to the synapse in optimal alignment. And so our hypothesis is that perisynaptic MMP9 degrades these protein bridges and reduces the efficacy of synaptic transmission. We're going through in a very targeted approach on Western blots, identifying specific components of these protein bridges, and then showing that light reintroduction after dark exposure increases the presence of a small carboxy terminal peptide, which is indicative of proteolysis, of cleavage of one of these proteins. But we're also very interested in testing the hypothesis that light reintroduction changes the strength of thalamic drive to the cortex. We're going to show that, we're going to test that 2 different ways. So in one experiment, we're going to use muscimol, which is an agonist for the inhibitory GABA receptors. And that is going to hyperpolarize cortical neurons. We're going to deliver muscimol to the cortex to hyperpolarize neurons. We're going to do it in the presence of another drug, SCH, which is a GABA B receptor antagonist that protects thalamic axons from muscimol. So thalamic axons have GABA B receptors. We want those GABA B receptors to be unimpacted by muscimol. And when you deliver muscimol and SCH together, you can isolate the thalamocortical component of the VEP because cortical neurons cannot spike. And that's shown here. So here's the LFP evoked in layer 4 prior to cortical silencing, the post-stimulus time histogram showing evoked activity. And here's a raster plot just showing activity along our array. So this is visually evoked activity. We're evoking spiking activity with our visual stimulation, and we're evoking this beautiful large amplitude VEP. When we add the cocktail to prevent cortical spiking, we do, in fact, stop spiking throughout the full vertical depth of the cortex, and we reduce the amplitude of the VEP, but we retain a small portion of the original VEP. This is the thalamic contribution to the cortical VEP, and then we wash out and recover. And following LRX, there's a significant decrease in the pharmacologically isolated thalamocortical VEP. Okay, so there's a decrease in the synaptic potentials generated by thalamocortical transmission. And then another piece of data, we're going to drive channel rhodopsin, light-gated opsin in the axons of the thalamus, and then post hoc in slices, we're going to look at the amplitude of the evoked EPSC, and we see that after light reintroduction and dark exposure, there's a significant decrease in the amplitude of the thalamocortical EPSC. So we have 2 pieces of data that suggest that dark exposure followed by light reintroduction does in fact reduce thalamic drive to the cortex. I'm going to show you 2 more pieces of data. When we use the FRET biomarker in live subjects, we can invoke in real time MMP9 enzymatic activity. And that's what's shown here. These animals are brought out of the dark and without vision. So they're having their first visual experience while they're on the 2-photon microscope. We're going to drive GFP in all pyramidal neurons so we can look at dendritic spines. And on the right, I'm showing the stability of the biomarker in the absence of visual stimulation in dark and in the presence of visual stimulation. So we get a very rapid and robust increase in biomarker fluorescence, which is reporting MMP9 activity. And the population data shows that this is significant across the population. But what's interesting about this is that the threshold for the induction of MMP9 activity is dependent on the history of visual experience of the subject. And so in animals that are not dark exposed, this is in blue, animals that are not dark exposed, the MMP biomarker fluorescence is stable in the absence of visual stimulus. But if we stimulate with 300 CDs per meter squared, which is roughly the luminance of the room that you're sitting in, we do not induce any enzymatic activity. Let's skip down to red. If we give subjects 10 days of dark exposure, however, and then give them the same visual stimulus, we see a robust increase in MMP9 activity that is perhaps at a plateau, because if we increase the luminance of our visual stimulus considerably to 150K CDs per meter squared, which is like looking directly at the noonday sun, okay, we do not increase MMP9 activity more than that. So we get a very robust, we lower the threshold for activation of MMP9 activity when we put animals in the dark, and this effect is blocked by the MMP9 inhibitor. I'm going to skip the final piece of data so I can talk a little bit about our efforts to translate this to humans. So the phenomenon that dark exposure reactivates plasticity in adult visual cortex has now been replicated by many labs and in many species. And so we use this as the foundation to apply for an R21 from the Eye Institute to get funding to run a pilot experiment to test the feasibility to translate dark exposure to humans. And this was done in collaboration with Ben Backus and Jose Manuel Alonso of SUNY Optometry. And we hired a program manager, Morgan Williams, and then we put 2 adult non-amblyopic males in the dark, and these 2 males are my colleagues. This is Ben Backus and this is Morgan Williams. So 2 of the PIs dark exposed themselves in order to report that this was very well tolerated, no reports of anxiety or boredom, no change in physical or emotional health. The Eye Institute was concerned about deterioration of mental health with prolonged isolation and visual deprivation. We measured a number of parameters during their dark exposure. Their sleep duration increased, but there was no phase shift in circadian rhythm. And this is very likely due to the fact that they had a very structured schedule in the dark. There's a team of people outside that provided meals and support and Zeitgebers that kept them on a normal circadian rhythm. We took melatonin. We took saliva samples twice a day so we could track melatonin, which increased in the morning and decreased in the evening. We took saliva samples for the cortisol. The most interesting thing about this initial pilot study is that both of the subjects report a chronology, a development, an emergence of visual hallucinations during their time in the dark. After about 28 hours in the dark, they begin to see phosphenes, individual points of light, that then turn into streaks, then turn into objects, and then become very complex 3-dimensional objects over the course of several days. Everyone that we put in the dark to date so far says that being in the dark for the long duration is not only tolerable, but it's enjoyable because the hallucinations are so entertaining. We saw no change in gross eye exam, no change in retinal exam, no change in visual sensitivity, including pupillary responses, stereoacuity, or contrast sensitivity. They were very photophobic for about 2 hours after coming out of the dark. They had an apparent enhancement of contrast and color and an increased pupillary light reflex following visual deprivation. Importantly, we documented an increase in the amplitude of visually evoked potentials that lasted several days. So this propelled us to run a small pilot in amblyopes, NIH-funded study, National Eye Institutes of Health NIH-funded study that we called Light Deprivation Utilized to Mitigate Amblyopia. And again, this is a feasibility test. It's a small cohort to test feasibility and safety in amblyopes. We recruited 7 anisotropic amblyopes. Again, anisometropia is a difference in refractive error across the 2 eyes, one good eye and one bad eye. And we first gave them an eye exam and corrected their prescriptions. Most people are not walking around in their best corrected prescription. So we corrected their prescription, gave them 4 weeks in best correction. And then 4 of them received 10 days of visual deprivation. Again, this was well tolerated. No reports of anxiety or boredom, no change in physical health, no lasting change in the visual system, including pupillary responses, stereo acuity, contrast sensitivity. Again, they were photophobic for 1 hour. They had the same sequence of visual hallucinations as the controls did. And we saw an increase in the amblyopic IVEP to high contrast checkerboards. We then sent them home with a virtual reality game and asked them to play the virtual reality game binocularly for 2 hours a day. And also gave them some homework, which was some non-virtual reality work, near work to do with a filter on their good eye to even out the acuity across the 2 eyes for dichoptic training. And here is the data from this pilot. So each of the 4 subjects is shown individually. Acuity is on the Y. This is contrast sensitivity function-based acuity. You can see prior to the dark, which is prior to their 10 days in the dark, which is the vertical gray bar, there's a big difference in acuity between the good eye, which is about 30 cycles per degree, and the bad eye, which can be as low in subject 7 as about 5 cycles per degree. But after coming out of the dark, we followed 2 subjects for about 150 days. We've followed the other 2 subjects for about 60 days. We see a modest, but nonetheless quantifiable improvement in spatial acuity in certainly 3 of the 4 subjects. So it's early, but we are very encouraged by this result. We're gearing up for a much larger cohort of amblyopes. So what I've shown you is that work in animal models, basic science, demonstrates that binocular visual deprivation can enhance plasticity in adult post-critical period visual system. Acute visual training is required following visual deprivation to promote the recovery of spatial acuity in animal models, and that visual deprivation appears to be well tolerated in human subjects with no adverse effects on ocular, physical, or emotional health, and we believe shows promise in the treatment of adult amblyopia, which is known to be refractory after the critical period. Just like to thank the people in my lab that contributed to the work, thank the people in the Bacchus lab that contributed to Project Luma, and also acknowledge my funding sources. Thank you.
Awesome. Thank you so much for that talk, Dr. Quinlan. We really appreciate you sharing your work with us. And we know, as I mentioned earlier, a lot of us, don't come from the bench science world. So you making all of this kind of accessible to us is something that I know that I appreciate and many people on this talk appreciate as well. So thank you so much again for coming in. We do have a question from Dr. Prakasa in the Q&A. She says, thank you for the excellent talk. Any role of learning new languages in older age improve neuronal plasticity and prevent dementia? What helps to activate neuronal remapping after stroke in adults, and any role for therapy there? I'm not sure if you can see that question yourself as well.
So the language question is very interesting. I think a lot about how to translate this to other sensory systems. One thing that we don't know, it hasn't been attempted, but one thing that we don't understand is what does the role of the internal dialogue play in constraining plasticity in language centers in the brain. But it's something that I talk to. We have a very strong linguistics group on campus, and it's something that we meet about and talk about quite a lot. One domain that we think is sort of ready for translation of this visual work is auditory. And the idea that individuals typically get, binocularly deaf individuals typically get one cochlear implant at a time, and you could induce a type of asymmetry in the quality of experience across the two ears by having those surgeries be asynchronous. And so I'm talking to auditory physiologists and clinicians about that as well. And we know, so it's appreciated that the time immediately post-stroke is probably the time in which the system has the highest level of plasticity. And often therapy, which we know improves function, is not initiated until long durations post-stroke. And so one area that I think is an interesting, fruitful one to explore is initiating therapy as soon as possible after the initial damage, because that's when the system is the most plastic and the most receptive to reorganization based on new motor commands that are coming in.
Thank you for that, Dr. Quinlan. We do have a second question as well. Are you able to?
I am.
Oh, perfect. Yeah. So I can kind of just allow you to look at it as well. It says, do you also see changes in the RGC spine dendritic density in amblyopic versus DE/stimulation?
So that's a great question. We have not looked. We've been very corticocentric. Now we're moving into the thalamus, of course, because we're interested in the thalamic synapses onto cortical neurons, and we will move back into the retina soon, or collaboratively. We predict that anything that changes in the retina, though, will be fully recovered after dark exposure and light reintroduction, because at the cortical level, we do see that full recovery of stimulus selectivity, and that's based on the input coming in from the retina, but we've not checked.
And we do have a third question. In mice, do you see an age cutoff where visual deprivation no longer works?
We do not, and we have pushed some of we've tested that hypothesis by trying dark exposure in animals that are about 2 years of age, which is about the end of a lifespan for a laboratory rodent. And we get very robust plasticity at that age. There's a series of papers by Kevin Duffy and Don Mitchell at Dalhousie that have replicated our work in cats. And they claim that there is an age cutoff in later adulthood in cats, but by their own admission, it's something I'd really like to explore. They're not doing physiology. They were using an anatomical assay, and so we don't know about recovery of visual perception or visual function. So there's a lot more work to be done in terms of asking that question across species, including humans.
Awesome. Looks like those are all the questions that we have in the chat. So again, just wanted to say, Dr. Quinlan, thank you so much for taking the time to share your work with us. We really do look forward to all the work that you do with Empower, and hopefully some exciting new collaborations between the translational research world and the clinical applications for them as well. So we'll be on the lookout for your name, of course, and we hope to have you back soon in the future for a talk where you can share exciting developments in your research.
Great, thank you.
Thank you so much, Dr. Quinlan. We appreciate it. Thank you, everybody, for joining. We'll make sure hopefully we can all see you next week for next week's talk. And until then, we hope you all have a great week. Take care. Thanks, Dr. Quinlan.
Bye.